And now let's continue with computational PTM proteomics, and Stefka, Tyanova will now give the tutorials on it.
So, good afternoon, everyone. Welcome back from your lunch break. And although it was highly recommended now to follow up with a siesta, please stay with me. I promise to try to stimulate your synapses in a different way. So, as Shivani just said, we'll now continue with another practical session of Perseus, and we'll be looking at phosphodata today. So, please locate again, your USB sticks. We need to go to the USB sticks and then software, Perseus, and just, again, double-click to start the program, like you saw yesterday with Tommy. And we'll immediately start with loading some data. So this is the green arrow here with the matrix upload. We click on it, then we need to navigate to our data. And in this case, it's again on the USB stick, data, practical 3 for today. And we'll be using the phospho-STY sites table. So this is the output table of MaxQuant when you have enabled variable modifications phospho-STY. It will then be generated, it will be there, and then this is what we recommend you to use for your phospho analysis. And then we open it. So you see that in this case, there are many more columns that we have in there as an output. We will not be using all of them, but in case you really want to see like your posterior error probabilities or scores, these are all there, and then you can use them and then plot to do some QC visualizations. We're going to go ahead and then take immediately the quantitative data that we're interested in. So just as a background, this is a fairly large phospho experiment that was done using really different conditions and different stimuli. We have control samples. They're indicated here with C, intensity C. So you would find 6 replicates, C1 to 6. There's EGF stimulation for 15 minutes with 4 replicates. So there's E15, 1, 2, 3, 4. And then there's nocodazole treatment, which simulates mitosis condition, again in 4 replicates. So now we're going to select some data to work with. And please pay attention as to what is important to put in there. So we will select 4 control replicates. So this is C1, C2, C3, and C4. And we're going to need all of these rows, C1 underscore 1, C1 underscore 2, C1 underscore 3. Then continue with the second replicate, again with underscore 1, 2, 3. The third replicate, underscore 1, 2, 3. And we'll just stop with 4, because we don't want to have too many, otherwise so the analysis will take too long. Then we'll move on to the EGF stimulation. Select the 4 replicates, so replicate 1 with the 3, underscore 1 to 3, replicate 2, underscore 1 to 3, and the 4th replicate. And we'll also take the mitosis condition. Again, 4 replicates, and this underscore 1, 2, 3. Okay. And we'll move them on the right-hand side under the main columns. One more thing that I usually recommend to be imported is this ID here. And the reason for this is that you will actually notice that all MaxQuant output tables have such an ID column. And this is usually ID that is specific for the particular table in which you find it. So in this case, we're looking at the phosphosite table. So this is ID that is specific for each of the sites. And what this can be used for is because, for example, in the other tables, you're going to have a column that says phospho ID column. Let's say in the protein groups table, you would have a column saying phospho ID. And then you can use this to match the two tables, because the phospho ID from the protein groups table corresponds exactly to the IDs from the phospho table. So, for example, in this case, you see that here we have the opposite. Here we have protein group IDs. It is already automatically selected in your case. And these will be actually the IDs from the protein group table. So you can use this always to match between the different tables. They correspond to each other. Okay, so there are other important columns that are automatically selected by the software. For example, you see sequence window, modified sequence. They're in there, and we'll look at what those are exactly in a second. And then there is another numerical column that is pre-selected, and this is localization probability. So Dan spoke about it in the theoretical part, and this is exactly the probability that is calculated in the software to know if your site is well localized. So what is up with, why do we have actually 3 different samples for just one replicate? These are the _1, 2, 3. So this actually corresponds again to these different quantification options that were mentioned at the end of the theoretical talk. And they correspond to the different multiplicity of phosphorylation at a given peptide. So if you imagine that you have a peptide that can be modified, that contains 2 serines and 1 threonine, so it has 3 potentially phosphorylatable residues. It's possible that then with the mass spec you actually detect different combinations of this modification. So it's possible that you actually once see a peptide that has just modification on the first serine, but it's also possible that you see a peptide that has modification on the 2 serines, or maybe yet another one where you see a modification of the serine and the threonine. So in this case, what you see is actually when you have _1, this corresponds to modification only of one site. So it means that you would know exactly it is the first serine, and this is the quantification that corresponds only to the peptide with one modification. _2 means that there are 2 modifications that have been observed. So the quantification of the first serine that comes from this peptide with both sites that are phosphorylated. And _3 corresponds to situations when there are more than 2 sites on the same peptide that are phosphorylated. And the idea behind distinguishing between these different modification forms is that you can actually argue that they may even have different functional relevance, right? You can say that, okay, if it's just one site modified, then the function of this one modification is X, but then or maybe it's, let's say, it's activating the protein, but if you have both of them modified at the same time, then it has an inhibitory role, just hypothetically speaking. But that's the idea behind distinguishing all of these different possibilities. Okay, so I hope you have all managed to transfer everything to the right-hand side, and then we just click OK to load the data. Yes. Well, no. Yeah, okay. ID is numerical, yes. I usually, okay, I added it under text. That's a good question, because just thinking about steps in the future, if you want to match between different tables based on identifiers, you would actually match text columns. So it is numbers usually, but because in the matching you match text columns, I put it under text. But that actually reminds me that if at some point you have loaded a certain column under a certain type and you realize you want to change it, you can do this. This is under rearrange, and there's the option change column type. So it's not a problem if you decide in the middle of the analysis you want to change this. And you see, if you click it, you can select any column type. So this means you already put it there, so head text. And yeah, you can just select any of these and then transform it to another type. So we're not going to do this, but just to know that this option is there. Okay, so You will notice that a lot of the steps that we'll do in the analysis of phosphodata are exactly the same as with the proteome. So again, we need to filter our table for the usual suspects, like reverse hits and contaminants. And I hope by now you know that it's the filtering are under filter roles. We'll filter again based on categorical column here. So we want to remove the hits from the reverse database. Just select it, we don't need to change anything, and click OK. Again, filter rows because we will want to remove all the contaminants. And again, this we usually do, but you don't have to because it could well be that you're interested exactly in one of the contaminants. So this is completely optional again. We'll remove them here. Okay, so the next step is actually phosphospecific, and that is because you will see that we have loaded this localization probability column here. Here it is. And as the name implies, it's a probability, so you'll see values between 0 and 1 here. And it is for historical reasons, I think, that people distinguish between different classes of sites. So we call class 1 sites, sites that have localization probability larger than 75%. So we usually recommend people to work only with those sites that have larger probability. Now, of course, you may have a legitimate reason to use lower localization probabilities as well. But we're going to go ahead and then filter according to this, just to retain class 1 sites. So we'll again use the filter rows option. But in this case, you see that this is a numeric value. So we'll filter based on numeric value. In this case, we have to select based on which column. So we said localization probability, and this is conveniently pre-selected by the software, but you see that actually you can filter on any numeric column, so you can use it for any other filter that you may want to do. Now here you have to specify how you want to filter, and here you have to write a small formula, and this is x larger than 0.75. So this just says retain everything that has a value larger than 75. And we do not need to change anything else. You can try.
Did you put it?
I did, yes. I did use spaces. Okay, so you can use it with or without spaces. The formula doesn't change anything. So you see now if you go to localization probability just to see if your filter worked, you can sort the column by clicking on it, and then you see that it's everything that has larger than 75% localization probability. Okay. Then next step that we did yesterday as well is to transform our data, which is under basic, transform. We can just take log2 or any other, doesn't really matter, we'll keep the default, and click OK. Okay, so the next step is again phospho site specific. So we have to do something about this _1 to 3. You see that we have here because these at the moment these appear like separate samples and experiments, but these are not. So this actually is the same site but just with different quantification. So we're going to use the modification specific menu, which is right here. And then actually if you expand it, you will find a lot of functionality that is PTM specific. In some cases only phospho when we're looking at kinase substrate relationships or linear motifs here, but in many other cases it also can be used for other PTMs. So now we'll use the first option, expand site table. And what this will do is actually to convert all these columns _1 to 3 into rows. So let's just go ahead and do it. So what you see now is that if we look at the table before, we had 36 columns. So we had 3 conditions, 4 replicates, 12 times 3 because of the _1 to 3. And after expand side table, we actually have all the main samples. So we have 12 main columns. And so the data was transformed. And then as an additional column, we got this multiplicity here, which is this underscore 1 to 3, so that we still know if our quantification actually comes from a peptide that was singly modified, doubly modified, or multiply modified. Okay, so the next thing we're going to do is we're going to define the different conditions that we have in our data. So specify our experimental design. And from yesterday, you probably remember that we can do this by adding an annotation row here. In particular, we're going to add a categorical annotation row. And we're just going to name all the controls as C for control. The EGF stimulated samples just with an E, so just have to make sure that all the replicates have the same group name. And the mitosis with an N. Okay. Okay, I hope you're all with me. So just continue. And then you see now we have our groups nicely specified. So what you will see when you look closely at the table is that actually it's heavily populated by NaN. So these are our missing values. You can really appreciate that there are a lot of this. And it's something that we expect in this case because our conditions are really very different. So the most extreme ones are, if we look at control and mitosis, because we know that there's really a huge number of proteins that get phosphorylated during mitosis. It's mainly regulated through mitosis. So we know that we'll have a lot of these missing values. So we can't really continue with the analysis. We have to do something about the missing values. So the first thing is to actually filter a little bit for the missing values. So we need to again go to filter rows and filter based on valid values. Now, here you really have many different options, specifically because in this case we have these very different groups and we know that the conditions are very different. So this is actually an ideal case to filter in a group-specific manner. So something that is harsh, but it could be a good suggestion is to require 3 out of 4, so that corresponds to roughly 75% in at least one of the conditions. So let's say this means that at least 3 out of the 4 values should be quantified in mitosis, let's say. Or even you can be even more stringent and say we want all of these values. And you can do this either by specifying the number of valid values, or you can also convert this into percentage and then set any percentage that you want to. And in this case, this would really be the recommended strategy when you know you have very different conditions, or for example, when you're looking also at pull downs or knockouts, it is really to filter in a group-specific manner and then be very stringent in one of the conditions because you really want to make sure it's not a fake hit that you're looking at. You're really sure about the quantification in one group and you afford to have missing values in the other that you can impute later. So in this case, so this is the recommended strategy. In our exercise, I'm going to do something different. I'm going to require 70% in total. And I do this because this is a much harsher filtering, and I do it just for the purpose of this exercise so that we end up with less data and we can do our analysis faster.
Short question?
Yes.
Over here. So how can it be that we already have rows with only nans from the very beginning?
Do we?
Yeah.
This means that, could it be that you didn't filter everything?
No, I mean, in the raw data, there's rows that are completely zero values. So how can that be if it's not modified? Why does it show up?
I think it's still possible that it's actually identified but not quantified. Because in this case, that's why we're also doing all this filtering for quantification. So it's anything that was detected as a modified, but I think there's no filter that says you need to have at least one measured value. Okay, so sorry. Filter rows on, oops, sorry. Filter rows, it's a very fast mouse, on valid values. I said for the purpose of this exercise, I'm going to say 70% in total. Reduce matrix. Okay. So as you see, we still have missing values, but by far not that many. But because in this case, we really want to use all of the data, and if you had done it actually in a group-specific manner, you would have to impute anyway. I want to demonstrate the imputation option. So it has a separate menu here in imputation. And there are, well, actually there are two different options for imputing data. One is replace missing values by a constant. This is actually absolutely not recommended, because if you just impute by a constant, you kind of enforce zero variance, especially in one of the conditions where you may have all of the values missing. And this is then going to really affect negatively your statistical analysis. So this is not recommended, it's still there. What we recommend is the replace missing values from normal distribution. So this is the option that we're going to use here. And you see that there are two parameters for this. There is a width and a downshift. So what this imputation essentially does is it draws a distribution of values that is, however, shifted to lower expression values with respect to your total distribution of a sample. So what this does essentially is that we make an assumption why we have missing values. Right? We say that we actually have missing values in our data because of the detection limit of the mass spec. They're really low abundant, and we can't see them and we can't measure them. I see a lot of questions all of a sudden.
Yeah, and yeah, we haven't done imputation yet, but I have actually done imputation in Perseus using this option. Mm-hmm. And usually, I mean, what I notice is that the intensity values that are replacing the nons are very low. I'm just thinking, it seems like the imputation method, as you said, it always assumes that it could have been detected, or it can't be detected because the intensity level is too low. But I think in reality, you usually have a mix of missing at random, meaning that, I mean, the DDA, something wasn't chosen, but it could be detected.
But exactly, in DDA, you usually go for the top n peaks, right? So you actually would, I think it's a valid assumption, but you're right. So whenever you have missing values, you have to know, this is a classic thing, are they missing at random? In which case, such an imputation wouldn't make sense. But because of the way in which we select this, and we kind of know that there is still this bias towards more abundant proteins that we see with mass spec, this is still a valid assumption for LFQ data. So yeah, I forgot to mention this is label-free data.
Because, yeah, I mean, the K nearest neighbor method is also very popular. And that sometimes I feel that it could have been a better option when I look at my data, because you can have, like, you know, intensity values in the range of like 20, 21, let's say, log transformed, and then suddenly the imputed values, there are like 16. I mean, they're far away from the the average if I didn't consider imputations?
This is indeed one possibility to use some sort of a machine learning method to impute this. The problem in this case is that you're already going to imply some structure in your data, right? So for the k-means, it already means that it will kind of have an idea of your different groups. So you'll be already imputing values, sort of helping to shape your different groups. And this is, again, something that you don't really want to do. You don't want to use any prior knowledge of what your grouping might be when you do this imputation.
Okay. Yeah, okay. Yeah. Just one last thing. It's just, I've read some papers on imputations, and the general, there is no really rule of thumb of which imputation method one should use, but in general, the missing at random imputation methods are doing better than the missing not at random.
Yes, but it's, as I said, you need to kind of know why you have this missing data. You're absolutely right. That's why I say you need to know why you're missing the data. But in this case, we work with this assumption. That's why we have this method. If for some reason you have different experimental setup where you think they're totally random, then yes, this is not the right method to use. Absolutely.
Yeah, thanks.
I think I saw more questions, but okay, they were all answered. Great, fantastic. So these parameters, the width and the downshift, they actually describe this random distribution and how it will be generated, from which the random values will be taken. So the downshift, this is actually the shift to the lower abundance. So obviously, the larger it is, the bigger the shift towards low abundance will be. So if you think that that's too big, you can always decrease this. Can actually put it to 0, then you can simulate even totally random values. And the width is really the width of the distribution from which the values will be picked. So we have some default values, as always, that are kind of empirically determined. We can stick with those, but so let's just stay with those. We'll click OK. But it's also really important to actually inspect the effect of your imputation. So we're going to do this. We can go to the histogram here. This is the shortcut. Or visualization histograms, either way. Select all of the columns. So just click OK.
Excuse me.
Yes.
Could I just please ask a question? Do we use replacement from total matrix or separately for each column? And what would be the difference?
So I think in this case it was from separately for each column. And again, in this case, it's probably a good idea because we know we have slightly different conditions. Okay, so looking at the distributions after imputation. You see the histograms here. So in order to visualize the effect of your imputation, you can actually go here to this selection from tab. It's quite hidden. And then you can't see it on the screen, it's too small, but it's essentially a selection from imputed data. So just click on this. And what you see highlighted in your histograms are all the imputed values. So you see, as you expected, that they're on the lower intensity side of each of the samples. And this is exactly what you aim. So as a rule of thumb, you don't really want to create a second peak, and you also want to stay somewhat within the main distribution. So it should not be a distribution that is lying really completely separately from your main distribution. So in this case, it's something that we could accept. Now, if you look at all of this, you would see that there are some cases that do not look quite nice. But again, these are one of, if you also go into the data carefully, you would see that these are some of the samples that have more missing values. And then you would expect this to happen. Okay. So we can go ahead and now do some statistical analysis, because This is roughly how we have prepared our data with imputation. We have already defined our groups. Just like yesterday, we have 3 different conditions. So in order to test for, in this case, differentially phosphorylated sites, we need to go to multiple sample tests. Under tests. And everything is essentially pre-selected, just like yesterday. We have just one group defined. We're going to use ANOVA, and you remember the parameters from yesterday. So we do not need to change anything again in this case.
Hello. So if I have a bimodal distribution, should I adjust a parameter to make everything normally distributed, or I just simply should happily accept those?
Correct. So because we say we have these empirical values for the downshift and the width, or I can just open it here. If you see that you create really a second distribution, then I would advise you to really change this downshift. Then you would have to reduce it to make sure that you remain within the main distribution.
But now you do have some of them looking like a bimodal distribution. Is that okay, or?
In this case, it's acceptable because it was just 1 or 2 samples, yes. You may want to normalize in this case. But again, it's a little bit trickier. So it's better if you can avoid second bump by playing with the parameter values. That is the right way to do it. So never create really a second separate distribution, because then whatever normalization you do, that's not going to help very much because you have just put way too much noise in the data. Play first with this. If you still think you may need some normalization, there's actually a function, a quantile normalization, that is really well suited for phosphodata, which is not yet in the version, but if you put maybe enough requests to Histogram, it will be there soon, hopefully, but that would be really the best way to normalize phosphodata. But yes, for now we can continue with this data. So we have done our multiple sample test. And it's really the same like yesterday. So we have the ANOVA significant hits, we have the p-value and the q-value. So now we'll really repeat the same thing from yesterday. The first thing will be to filter for only the significant hits. So go to filter rows, filter based on categorical column. And I'll be doing this just because I again want to prepare our data for the enrichment analysis. So we select the ANOVA significant column. In this case, again, we want to keep the rows that are marked with the plus. So we have to change this, don't remove them, but keep them. We click OK. So we are left only with the significant hits. And again, we'll try to define our different clusters, but before we can do the clustering, it's a good idea to normalize the proteins by z-scoring, just like yesterday. So it was a bit fast, maybe normalization, z-score. We don't need to change anything. Okay, our data is normalized. And then we can go ahead and do the clustering. We do not need to change anything. Can just go ahead and click OK. Takes a little bit longer this time because we have quite a few more data points, but okay, it's relatively longer. Okay. So nicely what you expect to see is again the separation between our different conditions. So you see the perfectly formed 3 clusters of control, EGF treated, and mitosis conditions. Now you're familiar with how you can play and then change the visualization here, but we're going to go ahead and just define a number of clusters. So this is this button here, define row clusters, because again, you remember that we want to be able to distinguish between sites that are going, let's say, up only in mitosis or up when they're EGF treated, and then to see what kind of biological processes are behind. So let's say in this case, will define 6 clusters. But again, there's really no optimal number. It depends on what you think makes biological sense. So we'll just take 6. You see they're defined everywhere. They're here, and again, just like yesterday, we're going to export those clusters with export row clusters. Okay, now to avoid yesterday's confusion, we're going to match back to the table before we filtered only for the significant ones, just to make sure that we have the complete data sets. So this is this matrix 10 for me. So before filter rows based on this was the significant column. So we'll click this one, and you probably remember that it's to match 2 tables, we need to go to multiprocessing, matching rows by names. The base matrix will be again the bigger matrix, that's the one before filtering. The other matrix is the one where we define the cluster. So to select it first, we click in this table, then we click on the workflow. It's selected. Click OK. Now there is a little trick here that I didn't mention. So how do we match the 2 tables and why there is a problem? So we can't use proteins because each protein can have multiple phosphosides. So then we wouldn't know what would be the one-to-one correspondence. We actually need to use this field here called unique identifier. And this is something that was created when we did the expand site table. Because you know that now for each site, we couldn't use the ID because we have 3 quantifications for each site, but they are going to have the same ID. So when we did the expand site table, there was also another column created, which is called unique identifier. And this is really unique to the site and to the specific quantification for this site. So we're going to select from both tables. We have this unique identifier. We know we have really the one-to-one correspondence. And again, the only thing we really want to transfer from the second table is this cluster column. So we're going to select this from the category columns. Okay, and we can click OK. Right, so now we have analyzed our data, we have defined our clusters, and we're ready to start checking if there are any functional categories that are enriched in each of the specific clusters. So first we need to load some terms. Yes.
Yeah, no. Okay, I missed the point where I transfer the clusters into the matrix. Do you know what I mean?
Where you export the clusters or where you match the two?
Exactly. When I have now the clusters, but in the heat map.
Okay, you have exported them, so you have a matrix in which you have the column cluster.
Yeah, but I missed the point how to export them.
How to match the 2 tables, you mean?
No.
Okay, maybe that's...
Okay, I have it now, yeah.
Okay, great. More questions at this stage? Okay, good. So we can again load some annotations, just like yesterday. So go to annotation columns here, and then add annotation. So this is actually HeLa. So again, the right organism would be Homo sapiens. We need to select the column to match the UniProt identifiers too. And it's a good practice actually to select the leading proteins column. And the difference between proteins and leading proteins is that leading proteins actually contains really the proteins that are identified with the highest confidence and with the largest numbers of peptides. So it could be the same or it could be a a little bit fewer of the identifiers that you're going to find within proteins. And then the rest is the same. You can just select, I'll select fewer of the terms that you're interested in looking at. So just like yesterday, the goal terms and the CAG. You click OK. Again, it will take a bit longer because you see that actually we have more than 23,000 rows. But it doesn't take that long. Okay, so we have the terms, we have the clusters. We can go ahead and do some enrichment analysis. This is in annotation columns, and we're going to use the same Fisher exact test as we did yesterday. Now, the big question is, why am I asking you to do the same thing again? If it's exactly the same, well, it's because it's not exactly the same. So we still want to check for enrichment in our different clusters. We still need to control for multiple hypothesis testing. We still need some thresholds, which in this case I will reduce because we said that actually we don't need to stick to 2%. We can change into anything. Just in this case, you need to know that 20% of what is reported in the final table may be things that are wrong or not with a high certainty. And now we reach to this relative enrichment point. And this is the really important part when you're doing enrichment in with phosphosides table, again, for the reason that one protein may have multiple sites that are phosphorylated. And when we're looking at these GO or KEG terms, these are actually annotation terms that are associated with the protein. So we know that we're talking about the localization of the entire protein or about the particular pathway that the protein is part of, and it's not a site-specific property. So we can't actually, if a protein has 10 sites, we don't want to count this 10 times, because this is going to then result in the wrong enrichment analysis. So this is the point where in the relative enrichment, we actually have to specify according to what to normalize this count. And you remember that we imported our annotation based on leading proteins. So this is what we have to say, to specify here. It's relative enrichment based to the proteins. And what this will do is it will count all the sites that are associated with this protein as just one. Or it will rather count the annotation term just once. So that is the only difference between phospho and protein enrichment analysis in this step. We can go ahead. This will again take a bit because it's just because of the size. So we have also 6 clusters, we have many categories, and then we have a very large number of sites that the enrichment will be happening. Okay, I see a question there. Anybody with a microphone? I see someone's coming. It's actually a perfect moment for questions.
Sorry. I was wondering about normalization. Also yesterday, it was done really at a later stage in the workflow. I'm wondering why is that?
Are you talking about this part?
Yes.
Yes, of course. So this actually you want to do only before clustering and only for the purpose of clustering. Because this is normalization across the different conditions of a protein. So you apply it only for this. If we were to normalize then based on the samples, this of course you will do much earlier. So you're absolutely right. It's just what and how we're normalizing. Yeah, thanks. Okay, more questions? Ah, there's one. Okay, I think maybe somebody can help you then directly with the problem that you have, will be easier. Okay, so we have the output here, and now just to make sure that you actually know how to look at the results. So we can go back to the, or you don't need, you can just follow on the screen. If we go back to our clustering, we defined our different clusters. Here we can just select one by clicking. So you see this is the last one. And like intuitively, you see that this is a cluster where it's mostly read in our mitosis condition. So this is a cluster consisting of sites that are upregulated during mitosis, right? And this is, so you see again, we have there are more than 8,000 sites in this cluster, and this is some random num identifier that corresponds to this cluster. So in this case, it's this 18229. And now we want to know what are actually the processes that are characterized by this pattern that are upregulated in mitosis. So we can go to the output table. You see this will be the enrichment for each of the separate clusters. So we can click again just to sort them. Or you could just filter only for this cluster, but I said this is this 2029 cluster. I just thought it would be nice to look at some of the terms that are enriched here. So what you see is spindle, kinetochore, regulation of gene expression, microtubule, cytoskeleton organization, and I know cell junction. So you already have the feeling that these are mitosis-specific ones, but just to be absolutely sure about it. Membrane, okay, that's different. Maybe I missed it already, but you would actually find the term mitosis here as well. So even if you didn't know what the experiment was, you could still figure out what was done based on this enrichment analysis, because this will really be the processes that are upregulated during this state. Okay, so let's go back to the matrix before we added the protein-specific annotation. This is matrix 16. And now we're going to go through the other modification-specific options, because there is much more that we can do that is PTM-specific. So, for example, we can add what is known as linear motifs. So let's go ahead and do it. And I'll explain. So there's one parameter that we need to specify, and this is actually the parameter name is sequence window, and there is a column sequence window, so that's already a good hint as to what should be in here. We click OK, and I'll now explain both. So in the table, you see this is the sequence window. So what this is essentially the modified or the phosphorylated residues, ST or Y, in the center, plus minus 6 amino acids on both sides. So it's not really a peptide, but it's the sequence window surrounding a modification site. And this again comes as an output from MaxQuant, and it's really very helpful because you can use it to match to many other external databases or to perform something like motif or sequence analysis. It's just there to use. And I think even so this data has been processed with a little bit older version, so it's plus minus 6 amino acids, but in your version this motif is even longer, if I'm not mistaken, plus minus 13 amino acids. There's another one here, just for information, is called modified sequence. Actually, it contains the peptide sequence and where the modification occurs. So you could actually know it's this third serine and the fourth, but not any of these two or this one or the last one. Okay, but now we added sequence motifs. So what did we do? Actually, there is one additional column here, which is called motifs. And what happened was that we use our sequence window, so this menu sequence window, to search for any known kinase recognition motif. So for example, you know that some of them do have this specificity. For example, at the first position after the modification site, there can be a proline, and this is one of the proline-directed kinases. So Perseus uses the sequence to match to motifs that are predefined. I can actually show you, and then it imports a hypothetical kinase that might be the one that has modified this site. So I actually see that for each site there can be quite a list of such kinases. And again, this is not a big surprise because a lot of these kinase recognition motifs are very overlapping. So if there is a proline in position +1, it can still be quite a few kinases. Or if it's an arginine at -3, again, there is quite a number of possible kinases. However, what we can do when we have these kinases here imported and matched is that we can use this also for enrichment analysis. And in this case, the enrichment analysis would be site-specific. And what this will tell us is that, again, if we, for example, had no idea what are the kinases that are involved in mitosis, right? We already know, okay, we have one cluster that has sites that are mitosis-specific. It's this one. So let's go ahead and then check which are the kinases that are potentially involved here. So it's the same thing. It's annotation columns, Fisher exact test. Again, we want to check for enrichment in the cluster or with respect to the different clusters. And in this case, we do not need to indicate relative enrichment because these kinases, they're site-specific. It could well be that we have the 5 sites on the one protein, but they're modified by different kinases, or they have different motifs, in which case it's okay to count them or as many times as they occur. So this is a site-specific annotation, and we do not need to do this relative enrichment. We click OK. We can again look, at the respective clusters or cluster, the one that we think is our mitosis-specific cluster. Here 29, and then you see things like PLK1 and CDK5 and CDK1, which are actually known to be related to mitosis. But in general, if you're kind of looking at a very novel condition and you want to know what are the kinases that are in there, this is a great way to figure out some of the kinases. Okay, so let's look at the other modification-specific options. So it doesn't really matter to which table you go as long as it's one of the main tables and not an output table of an enrichment test. I go back to this matrix 16 after the matching, but it's not really important. So in modifications, we have more options. We have known sites. We can go ahead and do it. We have to specify the type of modification. So we're looking at phosphorylation, the uniprot column, and again, the sequence window. So we need the information about the sequence. And what this will do is actually to use information from a great resource, which is Phosphocyte Plus. And it will check what kind of phosphosites in this case have been previously detected, either in high throughput experiments or in experiments that are specific to the company.
Excuse me, could you show the window again, and what did you select? It was a little bit too fast.
Oh, sorry. Actually, I didn't change anything. Maybe that's why it was so fast. So it's modifications, add known sites. As you say, also there's the little information, so sites known and from Phosphocyte Plus, add known sites. And essentially, you don't need to change anything here, right? It's the modification. So in theory, if you would be interested in a different PTM, you can specify this. Oh. Could it be that you actually don't see this? Why didn't anyone say anything? Great. So you don't see this because actually you need to place this information in the right place in Perseus. And because I did this before that, I didn't have any problems. So there's actually one right way to do it, which would be to go to the phosphosite plus site. You don't have to do it now, just this is how you would do it in future. The site is phosphosite.org. And this is an absolutely great resource where you find a lot of information, not only about phosphorylation, but about PTMs in general. You would have to go to downloads, datasets from PSP. For future, don't do it now. You will be asked, this doesn't usually happen. Of course it happens now. It's not our fault. But anyway, you would see a lot of tables that you can download for them. They're really very useful because there is information about if a site is known to have a regulatory role, for example, if it has been detected in previous experiments, or essentially any property that is known about this site from literature. So they actually go and collect literature, curate it, and then store all of this information in a really nice format. You can download this. We don't need to do it now because we have a really old version of this, but you're strongly advised to download it, to download the latest version because because we really have something that is quite old and we're not allowed to actually distribute it, so you'd have to do it yourself. But just for the sake of the exercise, you can go to the USB stick, Datum, annotation, and you would find a folder called PSP. So copy the whole folder. Inside you'll see there are many tables, but just copy the folder. Copy the folder and then go to the software, Perseus, bin, conf, and you want to paste the folder already at this level. So do not go into annotations, it's one level above under conf. Okay. So if you do this, if you have now the data there and with all the tables inside, if you go ahead and repeat what we did before, which was go to modifications, click add known sites. You don't really need to change anything here because we're interested in phosphorylation. And click OK, you should be able to see a new column which has the name known site. And essentially everything that has been reported in Phosphosite Plus will be marked with a plus. And there is some additional information if the site has been detected from a high throughput experiment or in a more targeted manner. So you would see that quite a large number of the sites that were detected in this study were already known, but also quite a few are novel. So I hope you managed to copy this data because a lot of the other modification-specific options here do require this data. For example, kinase-substrate relations. So as the name suggests, in this case, one would import, let's say, or map to the kinase that has introduced this modification, but the difference to the previous motif analysis is that in this case, this is experimental data. So it's not really just matching by motif, but it's a case where the site has been detected to be modified by this kinase. So we don't need to change anything. Click OK. And there should be a new column that is added. Phosphosite plus kinase. You can click to sort and then see. However, what you will notice is that we get information about very few sites. And as I mentioned, please go ahead, download the data for yourselves, and hopefully there will be more of these roles will be filled in. And again, you could, if you had more data here, you could do the enrichment using this column. And then this is even more specific because you're not kind of guessing what the potential kinase might be, but this is something that is measured. Okay, we can add one more of the options. This is, again, under modifications, add regulatory sites. It contains also very interesting information. It, again, comes from Phosphosite Plus. Again, we don't need to change anything. It just works with the sequence window and the proteins. Still have 10 minutes, right? Okay. You could, sorry, I just got distracted, but it's... So you see that there's actually a lot of columns got added after regulatory site. We sort, so the first thing is if it is known to be a regulatory site, yes or no, and then the really useful things are that in some cases you actually know what the function of this regulation is, which is really rare to have and great for phosphorylation data. So you know it's, oh great, it's in cell cycle regulation, that's good to see. Then you see if there are known interactions, which could also be very interesting. And then what is the effect of phosphorylation? And all of this can be used for enrichment, so they can tell you even if a process is up or down regulated. The problem again is that not so many of them are annotated. You would notice this. But hopefully as there is more literature published, this resource will also grow, and then you could use this information for the analysis. So are there any questions with respect to this? Because this is more or less, these are more or less the main things that I wanted to describe with respect to phosphodata analysis. You saw, okay, I see a question there.
I maybe missed, is there a possibility here to extract only kinases and phosphorylation sites on kinases which regulates their activity?
I think that will be not trivial because it means that you actually need to know what is the function of each of your proteins to begin with, if it's a kinase or not. So I guess you could use one of the goal terms for this, but it's not straightforward really to extract only kinases at the moment. There's just a list of kinases. Yeah.
Yeah. So you added all these modifications in different matrices?
There was no reason behind it.
Okay.
So you could add them on one and do the enrichments with everything together. Yes.
Okay,
I just
that
wanted
was my
to show you what are the specific columns that are added when you use any of these functions.
Okay, perfect.
Can this, some of these modifications that you have added, like for example the known sites or the regulatory sites, can they also be put for other PTMs, like ubiquitination, for example?
If it is in the phosphosite data, yes. So it's
like whatever.
Essentially exactly any table that you download from PSP and you put here in this folder, and you have this sequence window for the respective PTM, whatever it is, it will be matched and imported in the dataset. And you can just download the data and you don't need to change the format or anything like this.
For the enrichment analysis, what's the advantage of doing the clustering enrichment compared to 1D enrichment?
Compared to 1D enrichment? There is, I wouldn't say there is an advantage. So you can go ahead and use both methods. And actually, it could even be more interesting to do it with the 1D enrichment, because there you don't need to define any hard thresholds, right? You don't need to define your statistically significant hits, but you can do it on the entire data continuously. So you can go ahead and do it. However, with forceful data, it depends again if you want to do the enrichment on notations that are protein or site-specific, because if you do it for protein-specific annotations, you again have to make sure you do this relative enrichment analysis. And when you have continuous or numerical data, this is not trivial, because if you have 5 sites, which one do you take, right? Do you take the most strongly regulated one, the most strongly upregulated one, the most strongly downregulated one, the average? So again, there I would recommend the strategy of like splitting this continuous data into quantiles, let's say, and then doing the 1D with relative enrichment in each of the quantiles separately. So it just becomes more complex when you're looking at phospho data, and you need to carefully think about it. But you can definitely do both. It was a long answer.
Hello. In a very big question to the multiplicity column. How did you, or how was it used in the statistical testing, or can we do it for the later analysis for specific results for 1, 2, or multi-phosphorylated peptides?
So we essentially decided that each of these different quantifications will be kept as a separate event, so everything was tested separately. Because of this idea that maybe if you see just one modification on the peptide, it has one function, but when there are 2 sites, it plays a different role. So we just kept them as separate events.
Okay, so when you did then the clustering based on the rows, in one cluster there can be a mix of 1 time, 2 times, 3 times,
It
and
is
so on.
possible, yes.
Okay.
It is possible, though if you then inspect carefully your data, you'll see that these are not so many cases.
Okay, thanks.
So where do you define that this underscore that you were talking about is the multiplicity of the phosphorylation? And could you define it as underscore 1, 2, and 3, for example, all monophosphorylated peptides but in different positions? Or is this taken into account?
So this is the output of max quant. It already comes as 1, 2, and 3. And
but you could have more than
transformation.
Okay.
And if it's actually 1, 2, or more than 2. So 3 is kind of the average of everything else.
So every mono phosphorylated peptide
Site.
Yes, site.
Peptide, yes.
Okay,
Right.
in a different position is a different line.
Yes. Okay, I see no more urgent questions, but I'm happy to take questions at coffee breaks or anytime you manage to catch me. So thanks a lot for staying awake.